You're listening to the MER-Mates podcast, the marine science research podcast.
What is it really like to get a PhD? What steps are involved? How much traveling is required? If you have ever finished a master's and wondered about the next academic step, this episode is for you.
Today, we dive into the dizzying world of PhDs, with our guests providing an honest, no-nonsense take on the highs, lows, and everything in between.
But that's not all. Life in plastic is not fantastic. We also tackle a pressing environmental issue, microplastics.
Learn how scientists are measuring and tracking these tiny pollutants using living organisms like mussels.
Tune in,
Or should we say filter through,
this episode as we explore the behind the scenes of PhD life and the current research on oceanic micropollutants. It's one you won't want to miss.
Today, we have with us an environmental biologist specialized in ecotoxicology, who has a desire to improve the quality and health of ecosystems, focusing her efforts on coastal zones. She's based at PI, and she's a pre-doctoral researcher. Welcome, Nagore Blasco Bilbao.
Thank you. Welcome.
Welcome, Nagore. It's a pleasure to have you here. As Sam mentioned, you are currently a PhD researcher, but would like to focus a little bit more about yourself. So could you share with us how you ended up here in research?
Yeah, it's a very good question because I'm going to take away all the romanticisms here, and I have to be honest because I'm doing the PhD because I rejected the other options that I had in the moment. So yeah, the thing is that when I finished the degree and the master, I have a small crisis. I guess that all the people, all the students are in this time, So the thing is that I start thinking about what an environmental biologist could do, a part of the research, because in the university they focus a lot in the research and in the PhD. So I start searching for different companies here, and I found out that they are a lot of them, but they require you a PhD because they are high-tech companies that do research. So the only option that I had was the consultancy, environmental consultancy. So I have been working for 6 months, but I don't like at all. Yeah, I'm the unique biologist there. They were geologists and engineering workers, So it was a very different work. Was obviously a work that you have to do in the computer, a lot of legislations, writing in forms, and I think that maybe the PhD was the best option for me. And I know that I do the right choice because I'm happy now. Yeah.
Oh, well, we're glad you're happy. So it wasn't initially your first option, not at all?
No.
And exactly, so it was the computer work that you didn't maybe enjoy as much of environmental consultancy?
Yeah, part of that, you have a lot of customers. You go to sampling, but you go to an industrial zone, and you take soils. In the consultancy in which I worked, they do a lot of soil contamination stuff, so they take soils, but the analysis, the lab part, that it's the one that I like most, they did in another place.
All right.
So yeah, it was just to go to the customer, take the soil, and then write in forms. And another thing that I don't like is that they are your customers, so you have to get in line with the things that they want. I mean, sometimes you have to cheat a bit in the forms, and I don't like at all the.
So after that experience with the consultancies, you decided that the PhD was the best option at that moment. Tell us, how was the process when looking for a PhD, applying, or
Yeah, a long process. Also, I think that the one of the important or most important thing is to find someone with whom you're going to be working well. I mean, it's very important, the group in which you are going to work. So you have to choose the work instead of the topic. It's important, the topic,
Yeah.
but you can modulate the topic then, yeah? So I did the CTA here, so I knew Manu, and I talked with Manu, and we decided to apply the PhD to Plastemer project that was at time a project that it was starting. So I did all the bureaucracy with the Basque government, and yeah, finally I get it, the grant. Yeah.
That's quite nice. Yeah, you do have to go through a lot of paperwork.
Yeah.
Well, we don't really have the experience with a PhD yet, but for our masters, we did have to do a lot of paperwork, and it can be annoying. So now that you're here in Pi, well, you were already here prior to doing a PhD, but what does your day-to-day look like here?
Depending of a lot of things. For example, the first years you are quite relaxed and you do a lot of coffee breaks that are quite nice and really important. Yeah, thing. I still doing it, but not so much time. Yeah. But for example, now I am a lot of time in my desk with the computer, and I also go to analyze the samples because I think that the experimental part, the bioassays, so the experiments are quite short, but then you have a lot of samples to process, and a lot of your time is staying analyzing the samples, in my case in the microscope, and then processing this data in the computer. So yeah, depending on your PhD year, yeah, I think, yeah.
talking about all the time you spend sampling or in the lab, or even in the computer, what proportions would you say you spend?
Difficult question, yeah. I would say that the 70% in the computer. Well, yeah. I don't know. 70% right now. 70% in the computer, and 30% in processing. Well, processing or analyzing samples. Yeah. But in the first years, I also did the biomonitoring program for the PhD. So I was also in the in Urdaibai estuary, in my case. So I have different tasks. Yeah, now it's like only the computer and samples.
And do you prefer?
Yeah, I love sampling. Yeah, so yeah. But I would like to do a lot of samplings. I think that in my case, I've been a lot because some of my colleagues, they go like twice or something like that. In my case, we did during 1 year, 1 year and a half, and we go like all the seasons to sampling. So yeah, I have the opportunity to stay in the environment and taking samples a lot of time. So that's great for me. Yeah.
We hope to get to know more about all the sampling you're doing in Urdaibai later on. But I think it's something that we don't know. What year of your PhD are you on?
In the third one, third and a half. Yeah,
Out of how many?
I'm starting. Yeah, they are 4 years.
4 years.
Yeah, I'm starting the fourth in January of the next year. Yes.
Okay,
so,
okay.
Yeah.
And currently, what are you researching?
Yeah, and now we are going to start with the toxicological part of the project, because the thing is that we have the one first part of the project, as I mentioned before. Our group has little experience with microplastics, so we start the first step was to know and try to understand how the microplastics distributed in the environment and within the organisms in which tissues we can find them, to determine the target tissues and then try to find toxicity effects or something like that. Yes, so the first step, and during these 2 years, 2.5 years, 3 years, I've been doing experiments focusing on the distribution of microplastics. So once we did this first part, now we are going to start doing the toxicological part to try to know the effects that the plastic could have. Another thing to mention is that this first part we perform with commercial or model microplastics that are spherical, rounded, like perfectly rounded plastics. But the truth is that the plastic that we can find in the environment are not like that. Are fragments, they are aged plastics, so the toxicity could change. So the idea is to, in this new part, jump to these weathered microplastics and try to decipher the toxicity effects of the real plastics that we have in the real environment.
Quite interesting, but for our audience maybe, and even maybe for Shabby, because he doesn't know much, what exactly is a microplastic?
Well, but it's a difficult question because it's in the scientific community now, like, it's a debate, because the microplastics are considered, and the commonly expanded description, it's the one that the NOAA made in 2008, I think, and it's a small plastic particles that has less than 5 millimeter in the largest dimension. So this is the extended definition. But in 2019, Hartman published a paper in which he proposed to change a bit the definition of the plastics. So now, well, it's a debate, I repeat, but for example, for us, we prefer the second definition because they change a bit the range, and Hartman said that below 1 millimeter to 1 micrometer, it's the particles that we can consider microplastics, because in the other definition, they haven't determined the slower size of microplastics, were like less than 5 millimeters, and all of particles less than 5 millimeters were microplastics. In this new or adaptive description, it's like up to 1 micron. So we prefer to use that.
And you would have the nanoplastics, no?
Yeah, this is another thing,
This is another topic.
Yeah.
I know things.
Yeah, you do know more than what I thought.
Continuing with the microplastics, and nowadays more and more you can find news or in the public where why are microplastics considered as an emerging concern?
Okay, plastics, as you said, we can find more and more in the environment. We don't know the quantity of microplastics exactly that we have in the environment. So this is a problem because we know that they are all around the world. We can't find in prison zones in which the human can't live or is not living. So this is a problem, and we have a compound that it's a I don't want to say that it's toxic. Then, if you want, we can speak about that. But we have a compound very hugely distributed in the environment, and the effects are not very clear still. So I think that we have now a lot of efforts also to regulate them, but it's not something that it's controlled in the environment. So maybe because of that, it's still considering an emerging concern pollutant.
Well, I think you kind of more or less know what I'm going to ask next. And Well, you might not have the answer now. You might have to wait for that second part of your thesis. But why are microplastics considered pollutants?
Yeah, difficult question, because the truth is that the plastic is a polymer and is chemically inert, but the toxicity, it's because of the additives that it could have. Okay, so if you test the additives, you are going to have a toxicity. But if you have only the polymer, for example, the polymers that we are using are, as I told you, commercial ones are model ones, and they are just the polymers. So, well, we should know in the future, but they have not toxicity effects. Another thing is that, for example, the fragments, they have irregular shape edges, and they can cause physical damage in the tissues and in the cells. But the toxicity itself, it's because of the additives.
Perdonad.
The concept of if microplastics are pollutants or no. We will continue with the debate, no? But we will just close it and see what the audience.
We expect to hear your results soon
Yeah,
when you're done.
yeah.
Mm.
Another,
Yeah.
I can add something.
Yes,
yeah.
yes, yes.
Yeah, another thing that it's important of microplastics is that the smaller size, it's very important because it gives properties that the microplastics don't have. So, for example, they are so small, they have the proportion volume. Sorry, superficie volumen, ratio superficie volumen.
Ratio superficie volumen.
Yeah, volume.
Surface to volume.
Surface to volume.
¿Gancho desde ahí? They have huge surface to volume ratio, so they have the ability of absorbing a lot of contaminants, pollutants that are in the environment, and this is really harmful. Could be a really harmful thing because as our very small microplastics can be eaten by different size organisms, bioaccumulate and biomagnificate through the trophic chains, and if the plastics has in the surface another pollutants once in the digestive tract of the animals, they can detach and cause the toxic effects because of the pollutants that they have in the surface. Yes, so because of that, in our next experiment, we wanted to also evaluate the effect of another co-contaminants, metallic ones such as cadmium, and see also the known as Trojan horse effect, and evaluate the microplastics as vectors for the pollutants that I think, in my opinion, it's one of the most important things and the toxic things of the harmful thing of the plastics.
So microplastics don't harm only because of their presence, because they can also carry other pollutants, no?
Yeah.
Now that you mentioned animals and previously the environmental samples, when you refer to those concepts, what type of environmental samples do you usually use?
We, as the idea is to try to take the best photo of the environment as possible, we take samples from all the compartments, that's it, sediments, water, and biota. And within the biota, we take filter feeders as the mussels and oysters, polychaetes, shrimps, and another organisms such as crabs, that we can find in the beach. And the election of the organisms, it's because these organisms can't tell us which is happening in the compartments in which they inhabit. Filter feeders, they filter a lot of water, so they can accumulate all the things that are there. And the polychaetes, it's a very good indicator of what's happening in the sediment because it's organisms that eat directly on the sediment to extract the organic matter. So it's also a good indicator for us.
And from those organisms, which one seems to accumulate more, or do you have any results of which ones are most affected?
Yeah, we recently finished an experiment in which the objective was to try to know the distribution of the plastics in the environment, as I mentioned. So, comparing the different organisms that were in the microcosm experiment, that were mussels, shrimps, and polychaetes, we see that by large, the majority of the plastics were inside the mussels. Yeah.
Does that also imply higher side effects or more impact in their health?
Yeah, of course. Yeah, we are going to see in the next experiments, but yeah, because they can accumulate, and we also see that the smaller microplastics, 1 micron, can enter into the tissues. So, and they get trapped there. So this we think that this is going to be harmful for the muscle, and we will see what happen with the cadmium also.
So we know all about what you're working with. We know all about what you're working with, but we would like to focus on how you come to these conclusions. So what methods are you using with these environmental samples that allow you to find out all about marine microplastics interactions with your samples?
Okay, yeah. When you work with plastics, it's a bit difficult because you have no standard protocols to follow. You do a huge review of the bibliography, and you see different protocols with different steps. So it's a bit difficult, yeah. But in general, all the protocols have two different parts. That it's the digestion of the organic matter and the filtration, yeah, to obtain the plastics isolated in a filter. So in our case, we did some trials with different different type of digestions, alkaline ones. They are another ones that are acid or enzymatic ones, but we prefer and we use the alkaline digestion. And here it's also difficult because you have to be very careful to not degrade your plastics, because yeah, you need to remove the organic matter without affecting the plastic inside. So we use different reactive within the alkaline digestion, and finally we choose the potassium hydroxide.
Okay. I was wondering that, in my opinion, I thought that like plastics were so tough that you weren't even able to degrade them, but apparently you can degrade them.
Yeah, you can degrade it, and then when you are doing the characterization of the plastics, maybe the color is because of the reagent that you use, or the shape can change also. And then when you are going to do a chemical characterization, could happen that the spectrum that you should obtain is not the same that you obtain because of the acid or over the reagent that you use. Yeah.
And once you have this digestion done and you've got your plastics there, how do you quantify them? And is there a better way of quantifying?
Yeah, this is also, I don't know, it's difficult because the thing is that we are looking for very small microplastics. We work in a project with chemists, and they are interested in bigger microplastics because it's also easier to work in the lab. In our case, as we work with biota, we are interested in plastics that can interact with biota and can enter into the cells also to see then the toxicity. So for looking for such small plastics, we modify also the filtration step, and we do a filtration in tandem with different size pore filters. And this is also a problem because we use then the analytical quantification Raman spectroscopy, and it's when are very small, it's very difficult to have a result. Yeah. So yeah. Forgot the question.
Yeah, if the question was if there is there any better method for quantifying the microplastics?
Okay, yeah. In our case, we do a first screening in the stereoscope. I'm trying to quantify the potential microplastics visually, but this is a very difficult thing because you can't confuse with a shell or another thing or organic matter. So once I did the first screening, we go to Raman to analyze the plastic. The thing is that as we work with very small plastics and the filter are very small, yeah, the pore size of the filter is very small, we can't do a mapping with Raman that it's something automatically because it lasts very long time. So it's not a realistic way of doing it. So because of that, I do a first screening, and then the ones that I identify as potential microplastics, they analyze. But this is not something that all the papers did. I mean, they are studies in which they only do a visual identification, and in the environmental samples, we notice that we have more than 92 of overestimation of the plastics if you don't do any analytical verification. So this is a very important step that you can't miss, yeah?
What you explained, it's let's say a methodology that you and your team have created and designed. I could use, no, for
Yeah, yeah. We talk also with another partners that we have in Italy, for example, and they use more or less the same as we use. At, finally, as you have these two different steps, all the people did the digestion and the filtration, but you can change the reactive, you can change the incubation time, you can change the filter pore size, yes. So they are quite similar, but not the same one, yeah. And in our case, we read some papers that did things similar that the protocol that we apply here, yeah. So I don't know if it's I'm sure that we are not the only one using this one, but we did here the trials, and without following any protocol from one paper, we mix things from different papers, trying to obtain the best incubation time with the reagent that we think that it's the best for our samples. Yeah, because then depending on the matrix in which you are working, these times can be different. Okay, so for example, with mussels, with biota is a different protocol than from sediment, and the same thing, for example, with the stomachal contents of dolphin, for example, that we have here also.
You're also working with dolphins.
Yeah, with it's not for my thesis, but
It's just
it's in the project. Yeah, so I work with the stomach content, and for that we also need to change and adjust a bit the protocol. Yeah.
it must be very hard to keep changing the protocol and keep it
Yeah.
up to date and try to keep it relatively uniform with the rest of the literature going on.
Yeah.
maybe this is more anecdotal, but the last time that we're trying to find you here at PA, we're looking for in different rooms, and there was a dark room, and it happened to be that you were inside.
Pitch black.
Yeah.
Yeah.
Tell us what's going on in there.
Yeah, the thing is that the first part of the experiments that we performed were with the commercial plastics. So these plastics are fluorescents, so it's very easy to localize them. So we prefer to use them as was the first time working with plastics. We think that it would be better if we can find out the plastics like at the first view, yeah. So for that we use a fluorescence microscope and to see the plastics as are very 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 small, like a dot of. Yo sola me he metido aquí, ¿eh?
No me sale la mota,
Dust.
A dust particle.
Yeah, as dust particle. We need to switch off the lights because if you have light, you can't see anything.
I'm going to improvise a bit of a question.
Okay.
Yeah, because I was very interested at first when you talked about Urdaibai. Part of your sampling was going on there. And Urdaibai is just a fantastic place to be.
Yeah.
What exactly was your work going on in Urdaibai? What were you looking for? And do you have any results that's interesting?
Yeah, the idea is to try to know what's happening in a real scenario. Yeah, as Urdaibai, that it's a protected space. So we find two different sources of microplastics, and one is the wastewater treatment plant, and the other one is a company that manufacture plastics for car devices. So we think that maybe in the estuary they should be plastics. And of course they are, yeah. We only analyze the mussels and sediments, and in both we found quite a lot of plastics, yeah.
I didn't know there was a car manufacturing factory in Urdaibai. That's
Yeah, in Guernica, it's in the
Guernica,
upper
okay.
part, yeah. Yeah.
amazing. Well, we're reaching the end. Unless there's something you'd like to talk about and bring up. Maybe not. Okay, well, then we have to ask about a very cool part of the PhD, which I believe you should be doing soon, if you haven't already done, which is an international stay.
Okay.
So, where have you thought about going? Or if you've already been somewhere, why did you think about going there?
Yeah, I have to do next year, yeah. And we decide to go to, well, I'm going to go, I'm going to go to Portugal, yeah, to Aveiro, because as I mentioned, we are going to do the toxicological part with mussels, but we also wanted to do with polychaetes, okay? And here we work with polychaetes, but in Aveiro they have a lot of expertise with them. So they adapt a lot of biomarkers to the polychaetes, and we think that could be interesting to do something similar with these key organisms that we also use in Urdaibai to try to identify the plastics in the estuary.
How are you managing with Portuguese?
I can't say anything in Portuguese, yeah.
Regarding those international stays, it's a question more curiosity. Do you decide where to go because of the contacts between your university and the other university, or the director suggests you to go there, or you decide? Or
In my case, we decide based on the professional thing that we were interested in, but the most common thing is that your director suggests you going somewhere because he or she has contacts in the other place. In this case, the lab in which I'm going to go, I think that, well, my director, he has not the contact, but I think that someone from here went a long time ago. Yeah, so it's not an exotic place. Yeah.
I guess that if
Yeah.
I'd get, it's a pity that we don't get a say. Well, I guess you do get a say, but not as much as we would want to.
Yeah.
You maybe choose a sunny island in the Mediterranean, if not, or
Yeah, I would love to go to Azores.
Azores.
Yeah, it was my first idea, but the thing that the experiment that we are designing, it's better to do in the other place. So it's a pity, but
Well, you got Portugal.
yeah. Well, I like Portugal. Yeah,
Yeah.
good place.
getting to the end of the interview, something more personal and but more relaxed. What does Nagore do per time? What other hobbies do you have?
Well, during the week, nothing because I
Yeah.
have no time. Yeah. But well, yeah, I dance. I go to
All right.
dance. Yeah, I like a lot. I'm well now no, but I compete in hip hop group.
All
Yeah,
right.
yeah. So I like a lot dancing, and I try to go. Yeah, like when I can. Apart from that, I like to go to the mountain. I love nature. So I spend a lot of time there doing, I don't know, picnic, trekking. I don't mind. Yeah.
What's the best spot in Bilbao for a mountain picnic?
I love, well, it's not Bilbao, of course, but
Well, it doesn't have to be Bilbao.
no, here, yeah, like near, yeah.
Yeah, yeah.
Of course, Urdaibai, yeah.
Urdaibai, best place for a mountain picnic.
Yeah.
And do you have any book or scientific paper just for the audience? You need.
Okay, not about
It
microplastics.
doesn't have to be. Yeah, don't worry.
Okay,
Yeah,
it's related with biology at some point, but I like a lot a book that it's called The Invention of Nature, that it's written by Andrea Wulf, and it's a book that talk about Alexander von Humboldt, that it's a naturalist, a German, if I'm not wrong, naturalist that did an expedition into South America, and there he understand or he discovered a new way of seeing the nature and start connecting all the things that are in, for example, in a mountain, the plants with the climate, the organisms living there, the animals, and it's quite interesting because, well, this guy died a long time ago, yeah. So it was another time, and it's for me really nice to see this history from the beginning.
And we'll make sure that that our readers, well, not our readers, our listeners read it. Since you're a PhD student, we have to ask you this question because you're full into research, and you are like in the beginning of a research career. Well, apart from your work in an environmental consultancy, etc. Do you reckon that the work as a researcher is well valued or appreciated?
I don't know, a difficult question also. I think that it's not well valued because the salary, yes, because of that, yeah, and the instability of the contract. I think that in the university we have not the opportunity to work as scientists. You have to be a professor. So this it's very difficult to try to continue with your career because you have to go away, yeah. So I think that should be obviously because the scientific career, it's very difficult career because you have to be very focused, and all the people it's run, and it's a competence, yeah, all the time. So it's a sacrifice career, and we have to go away because here we have no opportunities. So a lot of people finish the PhD and go to as teachers to schools. I don't know. It's okay. It's
It's a good option.
yeah, it's a good option, but it's because they can't do anything else if they wanted to be here. So yeah, it's not good value.
It's a bit maybe an uncomfortable question, but what do you expect from the end of your PhD? What do you think will come after?
I don't know.
It's a mystery.
Yeah, it's a mystery. Another small crisis.
Another small
Suppose,
crisis.
yeah.
Well, we will soon have one too, eventually, when
Yeah.
we finish our master. But it's good to know.
I
Yeah.
think that this podcast also comes a little bit from a crisis, no?
It does come from a bit of a crisis, yeah. And being bored. Both.
Bueno.
Well, or getting distracted, just getting distracted with something else.
No, but just because we had the question like, what's after the master, so
Yeah.
we try to talk with being a PhD or has been a lot of years in research.
Yeah.
Discover more, no?
Yeah, and in PhD students, you have the question of what's after the PhD. So you could maybe start a podcast too.
Yeah, a good
Anyways.
idea. Yeah.
It's been fantastic speaking to you and learning all about microplastics and your work and your experience. We hope to hear from your results soon. And I hope that people are tuned into the Plaster MER project.
Yeah.
And that's all I have to say. Thank you very much for joining us
you.
today.
Thank you.